Welcome to the Jack Westin MCAT Podcast with your host, Phil Hawkins.
And Azaii Calderón Muñiz.
Hello. Welcome back to another episode. We are super excited to be here and to have you guys listening. Today, we're going to be talking about the cells that just don't listen. We're going to be talking about cancer and cancer cells and how the MCAT can test you on cancer, which is a very big, very broad topic, mile wide, inch deep, right? For the purposes of the MCAT, you will get tested on everything from different types of checkpoints, right, with respect to the cell cycle, its relationship to different ways that these cells are activated and how they can get away with not listening, how they can evade our immune system, but also you might be given information that you just don't know. And that's okay, because as we've talked about before, you can absolutely work your way through a passage with new information, use what you've already learned, and be able to apply it when you get the questions. So when you see something you don't know on cancer on the MCAT, don't freak out because you listened, even if the cancer cells didn't.
Right. I love that. I just kind of like taking a big step back for a second. You are probably going to get at least one, if not multiple passages on cancer. Like that is one of the bread and butter things that the MCAT does all the time. And so this is like something you really want to pay attention to. And just kind of like, once again, kind of taking a step back, looking at how the AAMC builds exams, like thinking about this, incorporating all of this information together to help you study better. The MCAT writers do not commission studies and pay for research because we want to have a passage about this thing. So we need to research this so that we can have a journal article so that we can have a passage about this thing. Like what happens is these passages already exist. These passages are already out there and the AAMC finds these passages and then writes questions to go along with it. So they make their exams out of journal articles that are already existing, which means that topics that are really highly studied are more likely to show up on the exam because there's way more journal articles. And so thinking about the things honestly, cancer is probably the most studied thing that you're going to see articles kind of put out about. I guess kind of depends on the year. COVID may have overtaken that at some point recently, but understanding cancer is a huge amount of publications over the last 20 years. And so if the MCAT writers are trying to build passages, there's a really, really good chance that you're going to have probably several passages on cancer. And as I said, they're probably going to throw stuff at you about some weird specific cancer that you've never heard of, that's got some weird, interesting pathway. And you aren't expected to know all of that, right? You're not expected to have memorized every type of cancer and every type of dysfunction that leads to cancer and the difference between a squamous cell carcinoma and a basal cell carcinoma, right? That's not something you need to have memorized the difference, but you do need to have a good understanding kind of background. And so a lot of the stuff that we're going to talk about today, some stuff might just be blatantly tested. They might just ask you questions about cell cycles. But I also think that this sort of thing makes it, understanding this really well helps you read passages better, which is, I think, where a lot of the problem comes in for students to deal with these things. Got really passionate there for a second. This is important.
It really is. And similar to how in the past, right, you and I have talked about how diabetes can be a physician's whole future career. There are specialties that focus on cancer. There are oncologists. There are folks that specialize in pediatric cancers, in hematology or blood cancers, right? And it actually brings me kind of to another really important point, making sure that we step back. There are tons of different types of cancer, right? But you can also have solid cancers, thinking, cancerous tumors, malignant tumors, but you can also have blood cancer, similar to how we talked about leukemia and lymphoma, right? The bone marrow for leukemia versus the lymph nodes and lymphatic system for lymphoma. And cancer really just, and I know everyone who's listening has this kind of basic understanding, just a reminder, right? Cancer is just your cells have stopped listening and they are out of control. They're replicating beyond what they should be, and they're going to cause more illness, right? Very rarely does a cancer just sit there and not cause any symptoms whatsoever. Although it can, but something to keep in mind as we move forward.
Right. And so we're going to kind of talk about this as like, first off, you can't talk about cancer talking about how cells normally grow, because that's a normal, healthy thing, is like cells replicating and growing and that sort of stuff. I mean, every single person here at one point was one cell, like you were a zygote, right? And so like that, going to the complexity that we have, like, with a full person with organ systems and different things there, like you have to have cells divide. And even then, we're going to talk about it, like there's cells that are dividing in your body all the time, and they need to be, and that's like a healthy thing. And if they stop, you would be dead. So this like cell cycle, which is what we call it, of this kind of like replication, it goes through like several stages. You have almost certainly heard about this before, but I'm going to reiterate it just because it's so critical and integral to understanding cancer. And so generally we go through this like cycle of growth where it goes G1, S, G2, and then the M phase, which is the actual like dividing into two. So G1 is a growth phase. Generally, think about what needs to happen for a cell to divide into two cells, right? Like it needs to get bigger, right? You want the cell to double in size and then split into two cells. And so both of the G phases, the growth phases, are really just about growth. You also need to make sure that you have the stuff you need before you're in the next phase. And so, for example, S phase is the synthesis or DNA synthesis phase. That's when you are replicating your DNA. So in G1, you need to make sure that you have got enough nucleotides to build an entirely new copy of your DNA. So G1 phase, you have like your cell gets larger, you get more organelles that kind of like, replicate inside the cell. You also like have to build more DNA in order to like synthesize new DNA in the S phase. Go to the S phase, you make a complete photocopy of your DNA. So now you got two sets. Then you go to G2, which is like that last little bit of growth that you need to accomplish. You need to make sure that when the cell splits into two, each cell is going to get like a mitochondria. Each cell like has everything it needs. And so you need to replicate a lot of the stuff inside the cell, but you also need to make sure that you're big enough that when those cells split off, they'll survive on their own. And then lastly, the M phase, where you're actually like going through mitosis, and there's stages within that we're not going to dive too deep into, right? That's like the prophase, metaphase, anaphase, telophase thing. But you actually have that kind of like splitting in there. One thing to note, so like if we look at certain parts of our body that are replicating all the time, like skin, intestines, things like that, they're going through that cycle, G1S, G2M, G1S, G2M, over and over and over, because they're constantly dividing. Other cells, like neurons, like those don't really divide that often, right? Or muscle cells don't really divide that often. And so a lot of times they're like outside this cell cycle, we call it, and they're just like hanging out in G0. So they're like, it kind of like branches off of this cycle. And so they'll just hang out there. You can kind of think about it as like the same as G1, only you're not actually growing. And so you're just hanging out. And so that's where you do have a lot of cells in your body that are in that G0 or G0 phase. But the cells that are constantly dividing are going to be going like G1S, G2M, just over and over and over.
Yeah. And mitosis is a great time that they're going to ask you not just about the cell cycle, but also, actin, contractile ring, tubulin, microtubules. So this, when they're asking you or when they present you a passage on cancer, right, they absolutely can ask you about these things. They can also branch even further. And so they might go one step separation, but the questions might go two-step separation, in the sense of, one step being asking about mitosis in general, and two steps being, the actual process of mitosis and the cytoskeletal components that are involved. And, this is another place they love asking about eukaryotic versus prokaryotic. They like asking about animals versus plants. And so they can ask you about the difference between, for example, the contractile ring in our mitotic cell division versus a cell plate, right? And what's going on in plant cells. And so these are all different possibilities that they can ask about with respect to or adjacent to a passage about a given cancer. And so that's really important to keep in mind. And like you were saying, Phil, there are cells in our body that do this all the time. And the cells in our intestines are actually pretty fantastic at going through this. They're very effective. And they can actually go through this process in a fraction of the time that a typical cell can do this. And if I'm not mistaken, and someone look it up afterwards, but I think it's like 24-ish hours or so for a cell to divide. And so if you think about the fact that your intestines can do it even faster, hopefully that makes sense, because you constantly have to have that brush border, right? All of those cells doing their best. Otherwise, if they're not at their best, you're not going to be absorbing your nutrients. And so that's a problem. This is also why something like chemotherapy, which targets a cell's ability to replicate, affects the GI system so much, right? You think about someone who's getting chemotherapy, and one of the first things that comes to people's mind is, the nausea and not wanting to eat and all of those things. And so that's why, the chemotherapy's effects, how we can understand the effects. And so there's so much going on, and hopefully that can, provide context for when future patients are going through this really tough time, and not just the patients themselves, but also their family and loved ones. We can think about, okay, in the future, not just how to address the cancers, but maybe there will be research on how to, target and continue. Actually, there will continue to be research on how to target it so that we can minimize some of the side effects while we're helping people, get to a better place as much as possible. So there's so much in this field of research that still needs to be done, but a lot of things that we can think about and use to just be able to connect with patients and remind ourselves of, okay, not just what they're going through, why they're going through it, and then with both of those things together, what we can do about it.
Yeah, that's a really good point, kind of talking about the nausea and stuff, because like understanding this cell cycle helps you understand not just cancer, but also how to fight cancer. Like once you know your enemy, like all of a sudden it's easier to kind of target stuff. And so most chemotherapies are targeting cells that are dividing and like messing with cells that are dividing. And we have cells that are dividing normally. And so those can be problems. And so that's why people who are on chemotherapy, right, like going through this, they have a lot of that nausea, right? We also talked a lot about how our bone marrow has lots of cells that are dividing all the time. And like part of that is white blood cells. So people who are on chemotherapy are like much more likely to get sick and have a harder time kind of fighting that. If somebody like gets a cut on their arm and they're on chemotherapy, it's harder for their body to heal that because cells have to divide and grow in order to like cover that wound and have that wound heal. And so this is like what's going on with like people who are on chemotherapy, right? Like you get bruises and cuts and they don't heal. You're like nauseous all the time. Like your hair, that requires a lot of kind of like growth stuff going on. So that's why your hair cells kind of like can't handle it and like you go bald. And it's because we're specifically targeting these really active or really replicating cells. But that's also why chemotherapy doesn't kill neurons for the most part, right? Because like they aren't dividing. And that's a good thing because the neurons wouldn't regrow. And so it's just kind of a useful thing to kind of like understand how all of that works together. But I also really want to kind of point out, because I know we're talking about a lot of basic stuff here, but like you said, the MCAT loves to ask like second order questions and like go a little bit deeper than that. So I think that like it honestly, like a great passage would be a passage about the machinery that builds like GTP and ATP and TTP that like builds like the nucleotides. And so they could have this big long passage about the actual mechanisms, the actual enzymes and how they work together. And, go into this thing about this anti-cancer drug that targets that machinery and is trying to mess with it. And then they'll ask you like what phase of the cell cycle is probably affected. And you need to know that like, oh, that's probably G1, because G1 is when we're trying to like build up enough nucleotides to actually do the S phase. So it'd be the G1 and S would be the areas that would be most influenced there. And so the MCAT will just expect you to put these pieces together. So even if you're reading a passage about guanine synthase, right? Like they're going to expect you to figure out how does this relate to everything else. And so you want to be putting these pieces together. They'll ask you like what other cells in the body are most likely to be affected by this, right? Your like bone cells, like let's say osteoclasts, they'll say something about neurons, they'll say something about skeletal muscle, and then they'll say something about the like lining of your intestines. And like that's the lining of your intestines is the one that's going to be affected by that. And that's like the intestines. I know we kind of talked like that brush border stuff, but like your intestines constantly, everything in your intestine is chewing up and degrading and breaking down stuff that we eat. And so that damages the lining of your intestines. And so that's why they need to replicate all the time. And so that's also why like colon cancers and things like that, intestinal cancers are kind of tricky because those cells are like dividing normally. They're dividing all the time. And so they have a little bit of a like more likelihood of kind of like going into that. But we also want to talk a little bit about telomeres because telomeres are really interesting. So basically what happens whenever we replicate DNA, because our DNA is in strings instead of circles, right? Like bacteria, their DNA is circular. And so like it goes and makes an entire like photocopy of that exactly. And so one plasmid is identical to the daughter plasmid that is replicated. But in I'm sure there's another term besides string-like, but in string-like DNA, where there's DNA in like strands rather than in circles, every time the DNA replicates, basically a little bit gets cut off the end and you like lose a little bit of that. And that basically kind of creates a clock where how many times can a cell divide? Because each time it divides, we lose a little bit of DNA off the end. And then it divides again, we lose a little bit of DNA off the end. And so we have this big stuff on the end of our DNA strands, like these big repeating segments called telomeres, which are basically just there so that we can cut stuff off and it doesn't damage like the important DNA. But after you go through so many rounds of cell division, then all of a sudden those telomeres, like you've cut them all off. And so you start cutting off the actual useful DNA. And so there's a lot of stuff going on in aging, but that's one of the main things that is studied because based on the way that DNA replicates, there's a finite amount of replications our cells can do going through this. Now there's some interesting things. There's an enzyme that can rebuild telomeres. It's called telomerase. Which is a great name for it because I love it when names make sense, but that builds the telomeres. And so that's active, like definitely early in your life and in certain cells, but it's not active all the time. But if cells are going to be replicating over and over and over and over, having telomerase active, like rebuilding those telomeres is going to help. Now, we kind of made it seem like, oh, we kind of want that because we want to like, we don't want to have a limit to the number of times our cells can divide. But remember that cancer is division of cells that's uncontrolled. And so in a lot of cancers, like telomerase is active and like you were actually like building that DNA longer so that we can keep replicating it and keep cutting off the ends and it's fine. And so understanding that, like if you could shut down telomerase for a certain period, like that would help fight cancer, right? If you can mess with anything in prophase, metaphase, anaphase, telophase, like the contractile ring, which Azaii mentioned in like telophase in humans, if you can mess with that, that's going to mess with cancer cells. And so you want to make sure that you understand kind of like all of these different facets that we're studying. And telomeres are definitely a testable topic. And they might like, honestly, understanding, like I could see the MCAT also asking a question about like why, like something about bacteria keeps replicating and like asking about like, what are the mechanisms for that? And there'll be an answer choice that's telomeres. And that's wrong because telomeres aren't needed when you have circular DNA. And so telomeres are not something that we see in prokaryotes, but they are something we see in eukaryotes. And that's an important thing to understand as well.
Yeah. And, like you mentioned, as you were saying stringy, I was like linear, but
yep, that's the right word. That's a much better word than stringy.
Yeah. And just kind of taking a moment because I think it's useful with enzymes. You see that ACE ending, you're thinking enzyme, and then the telo part makes us think telomeres.
Yeah.
So yeah, there's, and part of the other reason that we don't want these telomeres, or the telomerase infinitely active is inevitably, right? It's with the passage of time, with the replication of cells, the replication of the division of cells, the replication of DNA, our body accumulates mutations. It's inevitable. Every single one of you has new mutations that you did not, and changes in your DNA that you did not have when you were born, right? Hence their changes. And so these mutations and changes in our DNA that we've acquired with time are called somatic mutations. And so these somatic mutations can accumulate and affect important genes in our body, right? And so if the telomerase were infinitely active, you would just continue to collect these somatic mutations, and your increase of your chance of developing cancer, right, would go up. And because those would affect the important genes responsible for replication. And so pausing to think about this for a moment. Okay, so we've said mutations can promote cancer, but how, right? There are two main gene types that are affected in cancer. The first are called proto-oncogenes, and the second are tumor suppressor genes. So the proto-oncogenes, these promote cell division, cell growth, and so they are activating, right? They're activating your cells. And so you can think about this as a gas pedal. Now, tumor suppressor genes, they're going to inhibit cell division, and they're going to inhibit cell growth, right? They're going to promote apoptosis. And so these are the brakes that are normally functioning. Now, if you think about cancer, this uncontrolled cell division, you either stepped really hard on the gas, or your brakes are worn out and you really need a new set of brakes.
Yeah, you cut the brake lines.
Exactly. You cut the brake lines, the pads are worn down, you need to take it to the shop. But essentially these proto-oncogenes, if they get a gain of function mutation, meaning they are turned on more than they need to be, right? You've stepped on that gas pedal really hard. For the tumor suppressor genes, you want to think about a loss of function, right? You lose that ability to step on the gas. And so if you think about this, right, what types of mutations can affect these genes in a way that would promote cancer? So that's something else that the MCAT can test you on. So
Going
if you
into
think
the
about
genetics side of things.
Exactly. So if you think about a proto-oncogene, we want to gain a function. Would we want a nonsense mutation that's going to truncate or shorten the protein really early on? Probably not, because then you're less likely to have a gain of function if it's only a small part of the actual protein.
Yeah.
Doesn't mean it can't happen.
When you say, would we want that? We're talking about if we were trying to create cancer.
Correct.
Obviously, we don't want cancer, but.
Yes, but in a question stem.
Right.
So a nonsense mutation is more likely to be associated with a tumor suppressor gene mutation, right? Because then the tumor suppressor gene and its protein product can't do its job, and you've gotten rid of your brakes. On the other hand, a missense mutation might make more sense for the promotion of cancer with a proto-oncogene, because then you can have a mutation where, now this protein is even more active. Why? Remember what a missense mutation is. It's going to introduce a different amino acid that doesn't have the same properties as the original one. And so that might promote protein activation rather than inactivation. And again, this isn't perfect, right? But it's a general schematic to think about and a general framework to think about this with as you're working through a passage, but also as you're thinking about how the different topics on the MCAT tested on the MCAT Connect. And you've heard us say this a bajillion times, but the MCAT loves testing relationships. It's not just plug and chug, right? It's not just memorize. What can you regurgitate and memorize, right? It's what can you do with what you've learned, which is essentially what you're going to do in med school and your career as a future physician.
Yeah, absolutely. I really like that the gas and brake analogy, because that I think makes it a whole lot clearer. And a lot of times it's a combination, like your gas works 10% better and your brakes work 10% worse. And then there's another mutation that makes your brakes even another 10% worse. And like over time, you get enough things kind of going on where now this car is speeding way faster than it should be. But I also just kind of wanted to point out, we talked about how like a lot of our cells need to grow. This is just like homeostasis, right? Like our body, like all the time, our body's like, we need to do this, but not too much. We need cells to grow, but not too much. We need to raise our temperature, but not too much. We need to have salt in our blood, but not too much. And like, we need enough blood pressure, but not too much. And like, so we have these like homeostatic stuff where we're trying to push and pull, and like gas and brake is another way to kind of think about that. But applying that not just to cancer, but to other stuff, especially a lot of stuff in endocrinology of like understanding kind of like what's going on. A lot of times people get really confused. Like, why do we have these two different things that do opposite stuff? So like, because we need to push it, but not too far. And like, we need to have this ability to kind of like balance back and forth between those. And just in case, I think most people here are going to catch this, but like a missense mutation, that's like switching one amino acid for another, that could increase like the affinity for a product. That could also decrease, or a substrate. It could also decrease the affinity. And so missense mutations could really be in any, like the proto-oncogenes or tumor suppressors or any of those. But you're right, like nonsense mutations, way more likely in tumor suppressors, or frame shift mutations, like are way more likely. But you could also have mutations not in the gene itself, but in like the promoter region, or a mutation with some transcription factor that's increased. And so that increases transcription of this thing. And so it gets to be a mess, because like the mutations that cause like the proto-oncogenes to get more active, it might not actually be a mutation in the proto-oncogene. It might be a mutation somewhere else that is making you build more of those proteins. And that's like, you got more feet in that case, put that capable of pushing on the gas pedal. And so it's just kind of a little bit of a tricky thing, putting all of that together.
Yeah. And like you mentioned, you know, our body is constantly in this back and forth and walking essentially a tightrope. But it was funny. I was laughing because you said homeostasis, and two of our courses here are called homeostasis. So my brain immediately took me back to classes, and we covered this kind of stuff. We actually, and just as a frame of reference for people, some med schools at least, and including the one I go to, have a course on cancer that you're going to be taking, or have a section on cancer that you're going to be taking. That's how important this topic is. And so like you were saying, you know, that you really want to make sure that you know your different types of mutations and why some might be more common, right, in different cases. But you also mentioned this idea that it's not usually just one. And that's absolutely true, right? It's why we have this idea of the 2-hit hypothesis, meaning you probably need 2 or more hits or mutations in different genes for cancer to really progress or become a potential problem. And we were talking about how, you know, the cells in our gastrointestinal system just replicate and divide so frequently. And this is why something like colon cancer, or, you know, cancer in your intestines can kind of go unnoticed, but at the same time can be fairly common. And this is why we actually screen, right? Everybody has to get a colonoscopy at some point. Sorry to tell you. So they've got to take a look at your intestines. But it's because we know that at least some of these are slower growing, but they're pretty common, right? And so once we see it, we can take it out. We know that, okay, we've learned genetics at this point. Hopefully, if you haven't already reviewed genetics, go make sure to review it before test day. But we know that our genes, right, and all the alleles and the mutations that you were born with, right, those germ line mutations, they're passed down, right, and they're inherited. And so similarly, if someone already is born with a mutation in one of these genes, right, they're going to pass it down to their offspring. And now their offspring just need one more hit, so to speak, right? They just need one more mutation. And so we can develop these familial cancer predispositions, familial cancer syndromes. And you've definitely heard of it with breast cancer. You may have heard of it with colon cancer. You may have heard of it with different, you know, blood cancers and leukemias. And so there's this connection with genetics as well, not just with the actual genes themselves, but also in the transmission component of genetics that you can get tested on. But that's really important for us to be able to understand how we can target different cancers. And I remember I did some research a couple of years ago in a lab. And one thing that I was talking about with my my advisor at the time was the possibility of people who are born with these mutations. Can we take their cells, use CRISPR, or modify essentially their genes get rid of the mutation and give them back their cells, right? And it's this kind of thinking that is so powerful and could potentially revolutionize our ability to care for our patients. But you have to really understand what's going on at this very basic level to be able to have these kinds of conversations and for people to think about this and possibly make it a reality. And so there's a lot here. And just kind of rounding off our conversation on the genetic aspect of cancer, I also want to remind folks of some terms that we've talked about before, but the main one being incomplete penetrance, right? So this idea that, yes, you have the mutation, yes, you have the allele or the predisposition, but that doesn't mean you'll 100% get cancer, right? It's a predisposition. It makes it more likely. And so that's another term that they can throw at you on the MCAT related to cancer. And so there's so much, this is such a rich topic because there are so many connecting points. And like you were saying, Phil, there's just so much research on it because it's such a huge societal problem and burden. And I was looking earlier today, and the American Cancer Society says that at some point in their lives, 1 in 3 people in the US will get cancer. And so being able to really understand what's going on in addition to what to look for clinically, right? What symptoms to pay attention to, all of that is important moving forward as a society, as individuals, and as people who care about people.
Yeah, I do want to also, so I'm gonna share a little bit more. So I recently had a very immediate family member who passed away from skin cancer earlier this year, like a couple of months ago. It was something, we saw coming for quite a while because, chemotherapy and all of that stuff. But I just want to kind of like reiterate to people, like, this is something like it was, he was only like 23. And so this stuff can affect younger people as well. So definitely kind of pay attention to that. Cancer is fairly common as far as diseases go, like Azaii, as you mentioned, 1 in 3. I did a lot of research on skin cancer because of, kind of what was going on in my life outside. But kind of an interesting fact in Australia, because they had like the hole in the ozone layer and people are generally outside a lot more. 2 out of every 3 people will get skin cancer in Australia just because it's so like there's the sun is so much stronger there because of the ozone layer weakening that occurred in like the 80s and 90s. But their actual death rate from skin cancer is way lower because it's just so common, skin cancer, that everyone goes and gets checked all the time. And so understanding like how this stuff relates to our job as physicians, right? Like our job is to help find and catch these things and to educate the public. And like that's also a big thing that's really important. I also wanted to mention a little bit more on kind of like that 2-hit hypothesis. And that's why we have this incomplete penetrance. And we have this stuff is like, if I have like, let's say I was going to get breast cancer, and men can get breast cancer. Like I need to have like some mutation occur in a cell, right? And then I need another mutation in that exact same cell, right? And like at least 2. And so that's something that is pretty unlikely. Like if getting a mutation is like a 1 in 1,000 chance, like the odds of that happening twice to the exact same cell is 1 in 1,000,000. Like what are the odds of winning the lottery? And then what are the odds of winning the lottery twice, right? Like that's way, way less likely. And so in people who have certain genetics, like they need to get like something like that to occur. I need to get like 2 cells or 1 cell to get 2 mutations, at least 2, that cause this thing to kind of go forward. But with some people, they might already have one of those mutations messed up, right? And so in that case, like let's say I had that, like with the BRCA gene with breast cancer. Like if I had that mutation, then every cell in my body has one of those things already knocked out. And so if I get a mutation to any other cell, any cell in any breast tissue, period, that will like cause cancer, right? And so it's just one mutation to any cell versus like two mutations occurring in the exact same cell, right? Like that's way less likely. And so you can kind of think about it as like, what are the odds of somebody winning the lottery twice versus what are the odds of like in a city, somebody winning the lottery, right? Like that's way more likely. And so that's why certain people have like much higher rates of cancer in their family. That's why we do ask about cancer in our like screenings and our histories when we're doing our histories and physicals with patients. But that's also why like certain people, like if they know they have this mutation, it's really likely that they're going to get breast cancer. And so a lot of times women will just like have their breasts removed and do a double mastectomy as a preventative measure. Like they don't have cancer, but they're really, really likely to get it. And so at least compared to the normal population. And so like having that done is a way to kind of prevent that. But kind of like talking about that somatic versus germline mutation, right? Like if I like, let's say I got some mutation in like my colon, right? Like one cell in my colon has a mutation. I'm not going to pass that down to my kids, right? Because like that colon cell is not ever going to become a sperm or an egg. And like, so that's never going to get passed down, that genetic material. But if you have mutations that do occur in the germline, like if there's a mutation that occurs in the testes or in the ovaries, there's a chance that like that one cell got mutated in me. I turned that cell into a sperm and that goes to create a kid. And that kid now has all of their cells messed up because of that. So that's why germline mutations are way more serious than somatic cell mutations. Basically, you have germ cells and somatic cells. Germ cells are like ovary, testes, like sperm and egg cell. Everything else is somatic. And so it's important to kind of like think about that as well.
Yeah. And I know taking a moment and a step back, I know you and I had talked about it, but on behalf of the people that are listening, thank you for sharing with us and for being vulnerable and also for being willing and still being willing to have this conversation and do this podcast because that's not easy.
No,
but still
education is important. Is like the thing like, oh, if I can like talk to people and get somebody to pay attention to skin cancer, I think that is a good idea.
Yeah. And again, just on behalf of the people listening, but also again, from myself, just thank you. And you know, this idea of like the germ cells, like you were saying, it's so important. And like you were saying, you know, we ask patients all the time, okay, you know, does cancer run in the family? And sometimes people think, oh, does that mean that, you know, everyone has breast cancer, right? But there are multiple different types of cancers that can be associated with the same gene. And so that's why a good physician will also prompt and say, okay, who in your family has cancer rather than just does cancer run in the family? And people will often be able to tell you, and more often than not, you know, my patients and my loved ones and people I care about. Yeah, we can list a couple of people. And so it's important to get that, to have that thorough conversation. And that might mean for some patients that then they get genetic testing and then they find out, oh, I have a mutation, right? And they can inform their family members. And then those people can make more informed decisions, be on the lookout. Their providers can know, hey, we have to pay attention and we have to pay a little more attention than we might otherwise have paid.
Right.
Now we're going to shift gears a little bit and we're going to talk about what cancer cells actually do.
Yeah. Sorry. I got kind of like sucked in. So, cancer, super interesting topic overall. But like the thing with cancer is they've got something wrong where they are growing and dividing kind of on their own, right? Like they're growing and dividing. They have this like self-sufficient growth signal. A lot of times in order for a cell to grow, there's got to be signals coming from outside, right? Like, hey, we need more white blood cells. And so my bone marrow kicks it into gear and builds more white blood cells, right? Or like there's an injury to the skin, signals come in, say like, hey, we need to regrow more skin, dermis and epidermis and all of that stuff. And so a lot of times these signals come from outside. Now, cancer cells will grow without those signals, which is kind of a big thing. And they will not only just grow on their own without signals to grow, but they'll ignore signals to stop growing because those also exist. And so a lot of times, like people will, like you can get a tumor and that is not necessarily like dangerous, Because if you have like a cell that is just growing, but it's not like spreading, right? Like if you have like a fat cell that just keeps growing, and so you end up with like a big like thing of fat on your shoulder or your back or something like that, a lipoma, exactly. Like that's not necessarily dangerous, right? It might be something you want to have removed because it's like you don't like it, but it's not something that is inherently dangerous overall. Where things get dangerous is when cells metastasize and they start, like the cancer cells spread, right? And that's why we grade cancers the way we are. We grade them like stage 1, 2, 3, 4. Like that has to do with, are they spreading, right? Like, are they like aggressively going into other things? Because if I have cells, like cancer cells that are like, can, yes, grow uncontrolled, but also are spreading throughout the body, then all of a sudden I have this skin cancer that is not just like growing like a weird thing on my skin, but that's also growing in my spine and in my heart and in my liver and in my lungs and like all of these things. And so that growing is like starting to mess with the tissues. So like instead of just having like a wound on your hand, you kind of have wounds all over your body. And it's like fighting and killing and like breaking down the bone in your spine, or breaking down the walls of your heart, or breaking down your intestines. And so, or in the brain, right? And so like that's where cancer gets really dangerous, is all of a sudden when it's spreading and like starting to grow like in other places. And that's a really important thing, is to pay attention to, and like how those things grow. And so what's really interesting is like if I got a cut on my skin, right, or I damaged some skin cells, what happens is my body, my immune system will come in and kind of help like clear debris, essentially, is a way to kind of think about it. Like from like, oh, there's a bunch of dead cells here, we got to get rid of these so we can grow new cells. And so they will clear the debris, but we don't want to take big chunks of stuff and just dump it into the blood, because that could cause blockages and like the arteries and things like that, right? And so it dumps it into the lymphatic system, right? Which lymphatics is kind of like where a lot of our like recycling and is how I think about it. It's like recycling and trash and like debris from battlefields, like if we're fighting bacteria, all of that stuff goes into the lymphatics. And so our lymphatic system kind of like shuttles things around inside of our body. And so most of the time when cancer spreads, it spreads via lymphatic system, which is why we like very often when you have like cancer is spreading, the first place that it is going to start to grow a new colony of tumor cells, like that's probably going to be in the lymphatic system. And so very often you will see like through like a lymphatic chain where you have like 5 or 6 lymph nodes all kind of along the same pathway. Like one of them will have cancer, and then the next one will have cancer. And then all of a sudden it hasn't gotten to that one yet. Right. And so that's the situation you kind of hope to have as a physician, where you can kind of like see where it's going and remove all of that, like just get rid of all the places where the cancer is. So it is spreading, but it hasn't spread everywhere yet. And so there's different kind of like phases to this. Now, if it is spread to like all of your lymphatic system, you can't just go and remove all of that, right? Like it starts to become problematic. So you want to try to catch these things earlier. That's also why we like check lymph nodes, right? And want to be looking on that. I do also want to just side mention, there's a lymph node right on your clavicle here that we call the sentinel node, because it turns out that all breast tissue, all of the lymphatics drains into this one lymph node. And so we check that lymph node. We care about that lymph node a lot, because if you have breast cancer, but that lymph node is not like swollen because there's a tumor growing in it, then there's a good chance that the cancer is like contained to the breast tissue. But if it is swollen, that means there's a tumor there, and like the tumor may have spread a little bit farther. It's also an interesting one because you can actually see it. Like you can see in people like, whoa, that lymph node is really inflamed. We need to get you to a doctor ASAP and like check this out. And so it's kind of an interesting one, because a lot of lymph nodes we can't see with our naked eye, but that is one we can. And so that's one we care about a whole lot more.
Yeah. what's really interesting is those like lymph nodes in certain places that we know are more related to cancer actually have names. So like Burkhouse node and St. Mary Joe, like it's actually quite interesting. And that's why one of the things that they always tell us and you'll hear is check the supraclavicular lymph nodes. Basically, if you guys feel the sides of if you put like the fingers on the sides of your neck, you trace them down and you put your fingers in that divot between, the bones, your clavicle in the front and in the back, that kind of like little space and divot, that area is where you're going to be feeling. And when you're doing, like you were saying, and when you have someone with breast cancer, you're feeling all of their lymph nodes, their axillary lymph nodes, sentinel, right? You're going everywhere that's adjacent. And like you were saying, when we grade tumors and we grade cancer, what that means is we're seeing, okay, how far has it spread? Because we know that if it's gone to one lymph node, it can go further. And also it's important to keep in mind that the lymphatic system will eventually drain back into your vasculature, right? And so we can't guarantee that if a cell has, if there are cells and cancer cells in a lymph node, we can't guarantee that one of those hasn't escaped into the vasculature and metastasized elsewhere, which is often when, and again, I mention colon cancer a lot because the general surgeons, at our site do a lot of GI. It just so happens. And so I've seen a lot of GI, and that's a lot of what I'm exposed to. But when they're removing, a cancerous segment of the colon, even if there is a lymph node that is affected, they're going to recommend, and the oncologist, the cancer doc, is going to recommend radiation, right? Because we can't guarantee that a cell hasn't snuck past, the lymphatic system and into the vasculature and metastasized elsewhere. So these are all really important things that affect how we care and how we decide what intervention we're going to apply. Now, other things that these cancer cells are capable of. And I think one of the most shocking ones is basically this unlimited replication potential. They can recruit blood vessels and promote the creation of new blood vessels and bring nutrients in, right? Because they have this unlimited growth potential. They need a whole lot of nutrients. And so they're going to draw them in via angiogenesis, right? That promotion of blood vessel formation and extension. And this increased metabolic activity is the basis for some of the scans we can do, including PET scan, electron emission tomography, and see, how much, how metabolically active cells are and areas are. It also, and I want to pause a moment to think about something that many of you may have heard of, and many of you may not have, and those are HeLa cells. So HeLa cells are cells taken from a woman named Henrietta Lacks in the 50s. I think it was at Hawkins. And so 31-year-old African-American woman had cervical cancer. And at the time, informed consent for, research purposes and tissue collection was not a thing. And so this woman's cells were taken. And so they were used for research. And they are still used for research. That means over 70 years-ish of research have come from this one cell line, which is wild. This cell line, her cell line, has been used for, the polio vaccine, I think even the COVID vaccine, as well as leukemia research. And so this one cell line is essentially immortal, just keeps on growing. In this case, her family was not initially compensated. She was not compensated. I think in 2010, Hopkins started doing some things to honor her legacy. But this is an immortal cell line, which is wild. It has outlived the person by decades. So cancer cells, they really just don't listen. They have this unlimited replication potential for a couple of the reasons we've talked about, right? Including telomerase activation. That's definitely one of them. Now, they also have this ability to prevent cell death, apoptosis, right? Evade the immune system. Normally, our body will promote apoptosis when there is DNA damage, but cancer cells rely on this DNA damage. And so they need to be able to evade the immune system. And that includes evading certain immune cells like cytotoxic T cells. But they have all of these different abilities, and the MCAT will expect you to know the features of cancer and what cancer cells are able to do and what promotes their continued replication and, like you were saying, metastases, et cetera.
Yeah. And so I think some people might be like asking the question of like, wait, why were they using cancer cells to test vaccines? Like, why not use like normal cells? I think it's really important to like remember and understand, like in science, we don't want variables, right? Like if I'm studying something in my lab and Azaii is studying something in her lab and somebody's studying the same thing in the lab in China, or like, let's say I have this new breakthrough and other people want to see if they can replicate that. They want you want to have as few variables as possible. So the cells that I'm using are the cells that other people want to be using. We want to all be using the same cells so that we can all like check and make sure and understand. And like we have compatible stuff. And like that doesn't work in normal cells. That only really works in those immortal cell lines that will like growing and dividing kind of all over the place. I don't know how many labs Henrietta Lacks cells, otherwise known as HeLa cells, are in, but it's a lot, right? There's a lot of labs that have that. That's also why you'll start to like, especially when you are reading passages, I know for a fact when you start looking at the AMC exams and like practicing with those, you're going to see lots of references like, oh, we use the GM 1116 cell line. And like, that's that probably came from somebody and like might have been a cancer cell line. There's a good chance of that. And so like whenever you hear those like, oh, we use this cell line, we use that cell line, there's a good chance that it's actually a cancer cell line that we're using where just like something is off on that. Now, not always cancerous cell lines, but very often it is, especially when we're studying human stuff. Now if we're studying like mouse stuff, like we can breed mice to all have the same genetics and we can like kind of like amplify that genetics by like having like, oh, brother and sister mate, all of their kids are going to have the same genetics because they have the same genetics. And like you can start to do, I know it's kind of gross, but like that's way easier to control for in animal studies. But in human studies, when you're trying to control for like genetics, like you kind of want something that is going to keep working that way.
Also, to be very clear, there are not cancerous cells in the vaccine.
Yes.
Let me make sure to say that. I want to be very clear about that. So we use cell lines to test different things, right? Including vaccines. That does not mean those cell lines make it into the final product.
Yeah.
It is that what is being tested is put into the cells, right? To make sure that it will do what it needs to do. And then, right, it will be manufactured separate from that cell line.
Right.
I wanted to make sure there is no ambiguity about that.
Yes, exactly. That's like saying like, oh, I tested shampoo on a rabbit. That doesn't mean there's rabbit in my shampoo, right? That just means I put shampoo on a rabbit once to see if it like made the hair clean and soft and silky. So definitely, I'm glad that you do that because there's so much misinformation about vaccine stuff out there. But yeah, so another thing to kind of mention, and like this is once again kind of like taking a step back. There are lots of enzymes, kind of using the analogy where we talked about like brakes and like gas pedals, and about how like, you know, the proto-oncogenes like push faster and the tumor suppressors slow things down. I want to be clear here. It's not like one proto-oncogene and one tumor suppressor. It's like hundreds. Like you have hundreds of proteins that are like involved in like dividing more, dividing less. And so there's all sorts of these, but some of them are way more studied because of how they relate to cancer. Like we noticed that when this thing goes wrong, like you're really likely to get cancer. Like it really stomps on the brakes or cuts the brakes completely, or stomps on the gas or cuts the gas line completely. So those are things we want to study because if we can figure out how those work, maybe we can mess with it in cancer cells. And so there are certain players and certain enzymes that you are really likely to see, not just in cancer studies, but I've also seen like passages where they're studying like muscle growth, and they just like from exercising, like they're studying just like growing muscles, and they like, oh, you know, let's throw tumor necrosis factor in there and see what happens, right? Because we really understand tumor necrosis factor. And so there's a bunch of these enzymes that you want to be really familiar with. So TNF alpha is something that I've seen show up on multiple exams from the AAMC, right? It's not like it shows up. I've seen it once. Like it's something that shows up all the time. It's multiple times in the practice exams, and the MCAT's only released like 5 of those. And so there's lots of references to that. So that's tumor necrosis factor. Now that should make sense on what that does. Tumor necrosis. So this is something that kills tumors, right? And so this is something that stops growth, right? So tumor necrosis factor would be a tumor suppressor, right? Because it's something that kills tumors and stops cells that are growing too fast. And so it kind of like keeps track of the activity and activation of our cell cycle. And if it starts to go too much, TNF is going to shut it down. I will say I have seen passages from the AMC where they have referenced TNF and they just said like, we added TNF, and that's all they said, right? They didn't say it was tumor necrosis factor. They didn't say what it did, but they threw it into like here and made you try to interpret the graph. And if you know what tumor necrosis factor is, it's so much easier to read that passage and read that graph and know exactly like what is going on with this. So not only that, but TNF alpha can also do some stuff with the immune system because your immune system can kind of attack those cells. Other things involved in this, caspases. Caspases also cause apoptosis and kind of like kill cells when they're growing too much. They're proteases. But I think the important thing is like caspases and TNF alpha, you know that they are kind of pro-apoptotic. They're things that cause cells to kill themselves. That's actually one of the big defenses in our body against cancer. Like if a cancer cell starts growing too much, like there's like a bunch of these like little triggers or like traps in there that if a cell starts to grow too fast, the cell will automatically commit suicide, like via apoptosis, and it will get rid of itself. This is why it takes multiple mutations to develop cancer. It's not just one, right? Because even if you like, cut the brakes, like if the cell's got this like self-apoptotic like trigger, then it's just going to kill itself. And then obviously you don't have cancer because the one cell that had that mutation just died. And so regulating this is important. I also want to mention, I know a lot of times, or at least for me, I always kind of got confused between caspases and cyclins. So cyclins are basically proteins that go up and down in your cell cycle. Like there's some enzymes you need way more of in G1. Some you need way more in G2, some you need way more in the S phase or in mitosis. And so we have these enzymes that we have a lot more of during certain parts of the cell cycle, and cells that are constantly going through the cell cycle, like it'll go up and then it'll come down, it'll go up and then it'll come down. And so we have these cyclins, which are basically enzymes that are related to the cell cycle. Now, a lot of times cyclins like being messed up will lead to triggering some of these things like TNF alpha and caspases to cause the self-apoptotic stuff.
Yeah. And I want to pause for a moment because I find TNF alpha so interesting, because I know people are going to think, okay, so if it, is tumor necrosis factor, why don't we just upregulate it in folks who have cancer and use that to target cancer? What's really interesting is that when people develop cancer, the cells don't, the signals don't always work the way we expect them to. And so what's interesting is when people have done research on TNF alpha, they've found that yes, it is an anti-tumor, right, immune response, but it can also make tumors at some points less visible to certain cells of the immune system. And so in that case, it would promote the survival of those cancer cells. And so a lot of signals in the body can actually have in what seems like a paradox and contradicting fashion, or contradictory fashion, in some cases can be, cancer or tumor promoting or suppressing. And so it's really interesting and important to, again, we've pushed research, but that's how we understand things, and it's how we are able to decide what to do. And you guys are probably tired of hearing me say that, but I think it's important to hammer home. But there are so many instances where we go to, we think, oh, we know what this does, so let's target it. And then we realize, wait a second, we don't know everything it does, right? And TNF alpha is one of those. I think you already mentioned, but if not, maybe in my head, we talked about it, TP53, or you might just see it as P53 on the MCAT. And so this is something that is really interesting because the American Cancer Society says that there's an estimate of about 50% of cancer cells have a mutation in this tumor suppressor gene. And it's actually a transcription factor. So, it's similar to what we talked about earlier, and it's going to act at that G1 checkpoint. So it's going to prevent the cell from going into that S phase, that synthesis phase. So it's essentially going to affect DNA repair, right? Transcription might send the cell into that G0 phase that we were talking about earlier, might promote apoptosis, but it's a really important gene, and you're going to see it come up. I'm pretty sure we had already talked about it, but you're going to see it come up, if not on test day, in your career. Inevitably, it will come up at some point. And like we talked about, and I think I already mentioned it, but the T cells that TNF alpha can help hide the cancer from, those will normally, right, target cells that are replicating beyond what they should be, whether they are infected by a virus or they are malignant and tumor promoting, or cancerous. Those cytotoxic T cells, or CD8 cells that you might also see them be called, and that you probably heard us talk about in the immune system podcast, are really important. And so there are a lot of key players that we haven't even touched on, but these are a couple that you may very well see on test day. And as always, our goal is to make sure that you do the best you possibly can and succeed on the MCAT, but of course also succeed in the rest of your career and make a fantastic physician. And hopefully this has been helpful for you guys. It's been a productive conversation for us. And as always, you can head over to the Jack Westin website, the AMC outline. We have a lot of this on the outline. We have pages on mitosis, cell cycle. You'll see, cancer and tumor suppressor genes on there. You'll see TP53 at some point. And so there's so much on the website and so many other resources available to you. So if you haven't already, connect with one of our academic advisors. Their contact information is on the website, and dig into the resources. We're here to help you be the best version of yourself that you can be, both testing and beyond.